The genetics of asthma and allergy. It has long been appreciated that a predisposition to asthma and allergic responses runs strongly in families, suggesting the presence of an hereditary component. In addition, twin studies in humans and mice have implicated both environmental and epigenetic, as well as genetic factors in determining the susceptibility of an individual to hypersensitivity responses. With this degree of complexity, it is not surprising that the identification of the genes involved in controlling an individual's vulnerability to hypersensitivity responses has proven to be a difficult task. However, since the late 1980s, the genetist toolkit has markedly expanded with the availability of genome-wide sequence information in addition to libraries of single nucleotide polymorphism. These tools, along with more classical genetic approaches, have been used to map hypersensitivity susceptibility genes. One approach to determining which genes are associated with a particular pathological state is to use knowledge of the disease to develop and then genetically test an hypothesis regarding potential candidate genes. For example, we know that asthma is associated with high numbers of differentiated Th2 cells, and high levels of IL-4 expression introduce a bias in the differentiation of activated CD4 T cells towards the Th2 state. We can therefore hypothesize that asthma sufferers may exhibit polymorphism in structural or regulatory regions of the IL-4 gene, leading to usually high levels of IL-4 production. Using this theoretical framework, genetists selected a region on human chromosome 5q31 to 33, for detailed analysis. This region contains a cluster of cytokine genes, among which are the genes for IL-3, IL-4, IL-5, IL-9, and IL-13, as well as the genes encoding granulocyte macrophage colony stimulating factor. Careful study of this region revealed a polymorphism associated with the predisposition to asthma that maps to the promoter region of IL-4, a confirmation of the hypothesis advanced above. In addition, two alleles of IL-9 associated with a predisposition to atopy were also identified. A second approach starts with a statistically based search for genes associated with particular disease states and is referred to as a genome-wide association survey. The genomes of individuals who do and those who do not have the disease in question are mapped with respect to the presence of SNPs. Statistically significant association of disease with a particular polymorphism then provides the motivation for detailed sequence analysis in the region of the SNP and a search for likely candidate genes. Cloning of genes begins in the region identified by the candidate SNP and then proceeds by a sequential search of contiguous sequences until a gene of interest is identified. This technique is referred to as positional cloning, and several genes important in asthma and atrophy have been identified in this way. However, this investigation is still far from complete. Sometimes, the same SNP has been shown to have different effects in various racial or ethnic populations, illustrating the complexity of such disease-associated genetic studies.